Hello everyone, my name is Gabriela, I'm from Moldova and I represent Nicolae Testimițeanu University of Medicine and Pharmacy. I am thrilled to have the opportunity to share some of my research with you. I hope my presentation will be of great interest and offer some valuable insight. Thank you again for this opportunity, and let's begin with some basic notions about multiple sclerosis. To begin with, multiple sclerosis is a chronic neuroinflammatory disease characterized by disseminated demyelation in the brain and spinal cord, which means that the sheath of myelin around the neurons is slowly destroyed by unknown mechanisms. Clinically, it can manifest anyhow between blurred or double vision, trouble walking, muscle weakness and fatigue, to paralyzation, cognitive dysfunctions, and depression. This heterogeneity results from varying degrees of inflammation, neurodegeneration, gliosis, and repair mechanisms. The manifestations also depend on the type of evolution presented on the slide. Nowadays, the diagnosis relies on MRI, but MRI is not always sufficient for differential diagnosis. Other conditions can be similar when we talk about the symptoms, conditions that you can see in the left part of the slide. Therefore, early identification of multiple sclerosis remains a difficult goal to achieve. This is where laboratory biomarkers come. The introduction of biomarkers could facilitate earlier and more accurate diagnosis of multiple sclerosis, with potential to reduce the time of diagnosis and help personalize treatment. What we want from a biomarker is that it must identify people who are vulnerable to the disease or susceptible to a high risk of severe attacks, and predict which individuals are likely to show a response to treatment. Therefore, it must be rational, predictable, sensitive, specific, practical, and to depict a correlation to both the therapeutic outcome and prognosis. There are three types of markers that have been found: immunological, biochemical, and genetic, and I will be speaking about the most informative ones from these three categories. The first lab test that is informative is the genotypation of HLA genes. Polymorphisms in HLA class II antigens play a key role in determining the genetic risk for multiple sclerosis. Early research showed a positive link between the DRB1 haplotype and the frequency of multiple sclerosis. Some specific HLA-DRB1 alleles have been associated with an earlier onset of multiple sclerosis, which can be detected through MRI scans. Additionally, certain alleles like HLA-DRB1 0401, 0408, and 1601 are linked to an increased risk of developing neutralizing antibodies against interferon beta. This can lead to a poorer therapeutic outcomes for multiple sclerosis patients treated with interferon beta. Another marker that is pretty informative is the presence of oligoclonal bands of immunoglobulin G in the cerebrospinal fluid. These IgG antibodies are produced within the central nervous system and serve as indicators of inflammation. In patients with clinically isolated syndrome, the detection of positive oligoclonal bands in cerebrospinal fluid has been shown to double the risk of progressing to multiple sclerosis over a 4-year period. Increasing evidence emphasizes the significant role of intrathecal IgG and oligoclonal bands in multiple sclerosis pathogenesis. For example, actively demyelinating lesions are often associated with a substantial accumulation of immunoglobulins and complement activation products. Furthermore, the presence of oligoclonal bands has been linked to higher disease activity, increased disability, and elevated risk of a second attack that transition from a clinically isolated syndrome to early relapsing remitting multiple sclerosis and more pronounced brain atrophy. In clinical practice, oligoclonal bands in cerebrospinal fluid remain a highly reliable marker for diagnosing multiple sclerosis, highlighting the crucial pathological role of intrathecal IgG antibodies in the disease. Notably, oligoclonal bands do not correlate with other known central nervous system protein targets, such as myelin basic protein, proteolipid protein, or myelin oligodendrocyte glycoprotein. Another marker could be the MRZ reaction. The MRZ reaction, which involves antibody indices against measles, rubella, and varicella-zoster virus, is typically positive in most patients with relapsing remitting multiple sclerosis, but absent in other inflammatory neurologic conditions. The MRZ antibodies reaction in cerebrospinal fluid provides higher specificity for diagnosing multiple sclerosis and offers stronger prognostic value for the progression from clinically isolated syndrome to clinically definite multiple sclerosis compared to oligoclonal bands. A positive MRZ reaction, defined as a positive intrathecal response to at least 2 of the 3 viruses, was present in 78% of multiple sclerosis patients and only 3% of controls, yielding a specificity of 97%. The MRZ reaction was absent in 98% of patients with neuromyelitis optica or antibodies against myelin oligodendrocyte glycoprotein positive encephalomyelitis, 2 key differential diagnoses of multiple sclerosis. Furthermore, the MRZ reaction suggests a predominantly B-cell mediated immune response, which can help guide treatment decisions, steering clinicians toward appropriate immunomodulatory therapies. The potential pathogenic role of vitamin D in multiple sclerosis is supported by numerous epidemiological studies that have been shown a correlation between latitude, sun exposure, and the relative risk of developing the disease. Vitamin D has been found to suppress the Th1 immune response at multiple levels and facilitates the production of various neurotrophic factors. In untreated multiple sclerosis patients, lower levels of 25-hydroxyvitamin D are inversely associated with radiologic disease activity. Recent genomic research has identified a vitamin D response element near the HLA-DRB1 1501 gene, suggesting that vitamin D plays an inhibitory role in multiple sclerosis at the genetic level by interacting with this element. Interestingly, a study proposed that part of the therapeutic effects of interferon beta during relapses may be linked to an increased production of vitamin D. Research using animal models also suggests that vitamins beyond vitamin D may play a role in the development of multiple sclerosis. Vitamins A and E are thought to influence disease activity. Additionally, a small study of multiple sclerosis patients found lower levels of biotin in both cerebrospinal fluid and serum compared to healthy controls. There are so many biomarkers that I would need at least 2 hours to tell you about, and it would still not be enough time. For example, a high percentage of IgG antibodies against the Epstein-Barr virus protein epitopes have been detected in serum and cerebrospinal fluid samples from multiple sclerosis patients. In 2006, specific T cells targeting the EBNA1 epitope were isolated from multiple sclerosis patients. More recently, it has been reported that Epstein-Barr virus infection of blood-brain barrier endothelial cells triggers mechanism of disruption and the production of pro-inflammatory cytokines. The presence of Epstein-Barr virus antibodies is considered a marker of increased inflammatory activity and earlier disease onset. Also, both neurofilaments and tau protein levels are markers of neuronal damage. Neurofilaments are key components of the axonal cytoskeleton, composed of 3 subunits: light, intermediate, and heavy chains. In multiple sclerosis patients, light chain levels in cerebrospinal fluid are significantly higher compared to healthy control controls and patients with other neurological disorders, peaking approximately 3 weeks after a relapse. However, the correlation between the light chains of neurofilaments levels and disability progression is weak. In contrast, heavy chains appear to have a stronger correlation with disease progression, with notable increases observed primarily in progressive forms of the disease. Tau is a cytoskeletal protein primarily responsible for stabilizing microtubules. Elevated levels of tau in cerebrospinal fluid have been observed in multiple sclerosis patients. When tau and heavy chains levels are both elevated in cerebrospinal fluid of patients with clinically isolated syndrome, this combination has a 70% predictive value for conversion to clinically definite multiple sclerosis, which is more reliable than MRI in predicting disease progression. Based on all what I said, we can conclude there is no single biomarker currently that offers the ideal combination of sensitivity and specificity for diagnosing multiple sclerosis due to the pathophysiological complexity of the disease, which leads to a broad range of potential biomarkers. Continued research in multiple sclerosis biomarkers should focus on developing tools for earlier and more accurate diagnosis. This would enable clinicians to provide personalized, targeted treatments, ultimately improving patient outcomes and quality of life through timely interventions. Thank you so much for your patience and attention. Thank you to my coordinator and organizers for this opportunity. Stay healthy, safe, and curious.